And we know that it's just being projected like by 2050, actually, most, if not 50% and above, over a fish, for example, will be coming from culture systems and not from the wild. And we also know that the demand for fish is increasing. It is not going down, it is ever increasing. And yet we also know that our fisheries, some of the fisheries have collapsed, others have collapsed and are now recovering, others are really, really overfished. So that means they cannot meet the current demand. We have aquaculture where we have fish that is being cultured, and fish here, okay, let me say organisms, because we have fish, we have crustaceans, we have seaweed, we have all these things that are being cultured to try and bridge that gap between the supply and the demand. And it's the same one that I was saying. It's projected that by 2050, actually, aquaculture will be supplying over half of the demand for fish. The good thing with that aquaculture, especially, is there's room for expansion. There's room for expansion. You can culture more. It's not like the ocean when a fishery is depleted, then probably you have to wait for so long for it to recover, if at all it will recover. So I think our possibilities lie in aquaculture. That is where we can bank in. And because of that, then there is an interest. How do we improve our aquaculture? How do we develop new and improved breeds of farmed fish? Yeah. How do we make them reproduce better? How do we deal with the reproduction hurdles? How do we make them grow faster? How do we make them grow bigger? All those are questions. There's also the question of these fish that we are culturing. How do we keep them healthy? Yeah. And we know that there are fish vaccines. Okay, for those who didn't know, yeah, fish are also vaccinated. And there is a lot of hope there that through biotechnology, we are going to develop these vaccines, and some are already developed, and be able to apply them when needed. Then there is also the market, for example, for fish vaccine. You can see what the world is about 60 million euro, and is dominated by large healthcare companies, etc., etc. So the vaccines are already there, developed by biotechnology, and there's a lot of money in it. And who says that you cannot end up there? Now, the other application, or one of the things that is of interest, is fish breeding and stock management. Selective breeding is a recent activity in aquaculture. If you remember in SMR 301, I told you aquaculture lags behind when it comes to breed improvement for so long. When this technique has been applied for so long in animals, in cows, in goats, in sheep, in all these other terrestrial animals, aquaculture lagged behind in trying to improve the fish breeds, probably because the interest was also very low. But now there is quite a bit of that, the selective breeding. And there is a lot of promise in that, and there are ongoing genetic enhancement programs that are targeting improving the fish breeds, their productivity, and this includes mollusks, it includes crustaceans, and the rest. Now, there's also the use of molecular markers to enhance selective breeding. It's like, what do you want? What do you want to pass on? Do we have that trait that you want to pass on to this other whatever organism? Can you select? Can you use molecular markers to check for that trait that you want to pass on? Are you sure it is there? And can you pass it on maybe by crossbreeding, by hybridization, and that kind of thing? Then of course now we come. So remember up here, this is still non-gene technology, biotechnology. Yeah. But when you come here to the genetically modified marine organism, then that is gene biotechnology. You're going all the way down to the gene. Now, genetic engineering is considered to have significant potential to enhance aquaculture through the use of course, recombinant DNA techniques that would yield faster growing and or disease-resistant varieties. So that is already being looked into. How can we grow faster growing fish? And you can imagine if you're growing tilapia for 5 months, and you can shorten that by half, you can almost imagine what you'll be saving. So if you look at both plants and animals, there are some key similarities, okay? And this includes both in the terrestrial and also in the marine areas, okay? So there's always the issue of reproduction, successfully ensuring that the plants or the animals have been able to reproduce. There's also always the issue of nutrition. For plants, how do you enhance their nutrition? For, of course, the same with the animals. There's also always the issue of growth. Do you want to grow them faster? Always is that we want the plants, we want the animals to grow faster than they are. And of course, there's always the issue of health. That means all these are areas of intervention, areas that we can use biotechnology to intervene and improve the culture of these animals and plants, and in the process, improve productivity. So I think before I go to that now, let's now watch the video, which I skipped earlier on. Let's see, let's just get an introduction to marine biotechnology. Just a minute.
So what do we understand by marine biotechnology, which is also known as blue biotechnology? A simple definition is the application of biotechnology.
Can you hear me? Can you hear? Can you see my screen?
Yes.
Yes.
Am I the only one not getting a what?
What's the problem? Is there anyone talking in the background?
We ain't seeing the video.
Sorry? Just speak clearly. Are you able to watch the video? But there are some people who say they could see. That's why I always ask. And when I ask, people say they can see. So let me upload.
We saw the net post.
Okay, let me upload again. Can you see my screen?
Yes, you're seeing.
Can you see my screen?
Yes, you can see it, but
Yes. But it's not playing.
it's not playing.
Hello, and welcome back to the Blue Generation MOOC. In this module, we will talk about the sector of marine biotechnology, a very new and important sector. So what do we understand by marine biotechnology, which is also known as blue biotechnology? A simple definition is the application of biotechnology on living marine resources. This means that the sector will be using renewable marine life forms such as fish, algae, and macro or microorganisms to produce a very diverse variety of new products that include food products, animal feed, medicines, cosmetics, and even energy. Marine biotechnology is therefore partly overlapping the sectors of fisheries and aquaculture due to the fishing extraction of the bioresources. But it sets itself apart from these sectors by developing new products and services using a variety of marine life forms. Data provided by the industry claims that the EU algae biomass sector currently employs 17,000 people in both direct and indirect activities. Annual turnover was estimated at 1.5 billion euros, with an additional 240 million euros in indirect activities, which include research. This sector involves some 560 companies and more than 300 research groups throughout the EU. For the entire marine biotechnology sector, it is difficult to determine its exact economic value and employment, as it encompasses a wide range of activities and is still a very young sector. The total employment in the EU is thought to be up to 40,000 people. These are usually high-end jobs and are the result of substantial public investment in education and training. The main applications of biotechnology in the EU economy fall into the following groups. Firstly, there are healthcare and pharmaceutical applications. Marine biotechnology has led to the discovery and development of a range of advanced medicines, therapies, diagnoses, and vaccines. For example, biotechnological breakthroughs have created new medicines for patients suffering from metabolic diseases, multiple sclerosis, rheumatoid arthritis, cancer, and Alzheimer's disease. Secondly, in agriculture, livestock, veterinary products, and aquaculture. For this subsector, biotechnology has improved animal feed. It also has produced vaccines for livestock and is improving diagnostics for detecting diseases such as BSC, foot and mouth disease, and salmonella. It has also enabled the use of enzymes for more efficient food processing and improved the breeding of plants to obtain desired characteristics. Thirdly, in industrial processes and manufacturing. Marine biotechnology uses enzymes which through biochemical reaction are improving the production of detergents, pulp and paper, textiles and biomass. This improvement can mainly be measured in form of a lower energy and water consumption and also a lower production of toxic waste. Fourthly, in energy production. Marine biotechnology is using micro and macro algae technology to produce energy, with microalgae being single-cell microscopic life forms and macroalgae being large aquatic plants such as seaweed. It is estimated that a theoretical volume of 20 to 80,000 litres of biofuel per hectare per year can be produced. Worldwide algae biomass production has tripled in the last 20 years to a volume of about 30 million tonnes per year, of which China was producing 47% in 2016. In Europe, the production is still low, with the EU only contributing 0.2% and the EEA contributing 0.8% to the global production. Norway, France and Ireland are the main European producers of algae biomass. And finally, in daily consumption products. Here, marine biotechnology has successfully developed new food supplements. Examples are proteins, fatty acids, probiotics, vitamins and minerals. It has also had a big impact in the cosmetics industry with products including moisturizers, vitamin C complexes and anti-aging formula. Employment opportunities partly overlap with the aquaculture sector, as often marine biotech companies have their own aquaculture fields of macro or micro algae. Therefore, also jobs that can be found in the aquaculture sector are available in marine biotechnology. Examples are biologists, aquaculture technicians, engineers, shuttle boat skippers, divers or farm managers. As the sector is emerging, it currently offers more high-end jobs on the research side, such as microbiologist, wildlife biologist, biomedical engineers, and medical scientists. But marine biotechnology is developing and expanding, and therefore also the production end will be further expanding and offer employment opportunities to marine harvesters, laboratory staff, and mechanical assistants. The expected employment growth is about 1,000 jobs per year, but as the sector is fairly undeveloped in Europe, there are many possibilities also for small SMEs and startups. What this sector is looking for are young people with self-motivated personalities and a passion for long-term goals. Also sought after are the ability to develop challenging ideas, fail, be wrong and rebuild from lessons learned, the ability to explain complicated concepts to non-experts in simple terms, an understanding and elementary knowledge of business development, economics, business planning, cost planning, logistics, production, upscaling, and market analysis is also valuable. In this young sector, there are only a few new and focused qualifications available. One is the Blue Biotechnology Master, offered by the University of La Rochelle, which lectures on blue biotechnologies and is dedicated to their application in the health, nutrition and aquaculture domains. The University of Tromsø in Norway, is also offering a 2-year master degree of marine biotechnology that teaches biotechnological expertise with particular emphasis on marine resources. Many other universities, such as Wageningen University in the Netherlands or the University of Ghent in Belgium, include modules of marine biotechnology into their studies for different master degrees. To a lower educational level, pathways such as laboratory assistant or mechanic can be an entry qualification into this sector. Overall, the sector of marine biotechnology offers new and exciting opportunities, and as a growing sector, holds a large variety of good future career perspectives.
Okay, back to class. The reason why I chose that video is just because of the fact that not only does it give you an outline of what marine biotechnology is about, but it also tells you about the opportunities that are there. And I think you've had it for yourself. The opportunities for young people who are self-motivated, who think they are creative, who are ready to try something and fail and try again until they get it right. Okay? So, and the fact that they've also said the fact that he's named some of the universities that are opening, that have such opportunities. I think the University of Ghent in Belgium also offers. We've got quite a number of our KU students who have applied for the VIA scholarship, and they've gotten it, and they've gone ahead and studied abroad for their masters. And most of them are doing very well. Actually, those that have come back, they are doing very well. So I'm just doing all this to provoke you to start thinking about what you want to do after this. You may actually not even decide to pursue a master's degree or that kind of thing. But how can you apply? Do you think there are ways of applying some of this information, this knowledge, into something that you can self-sustain yourself? Like, let's think the issue of seaweed farming. It's one of the marine biotechnologies. We're using seaweed for all those things. There's a lot that people can do with seaweed. And yet we're not seeing that. We have a lot of seaweed in Kibuyuni that is just lying there waiting for people to buy it. What can you think? What do you think you can do with it? Okay. We're starting with small SMEs. If you put your brain together in knowledge, what can you come up with? So I think the opportunities are out there, but we need to start thinking outside the box and look around and say, what is available here? What can I do with the knowledge I have, and with the more knowledge that I can get? What can I make? And you realize that funding may not need anybody to employ you. Okay, not only will you be employing yourself, but you'll also be able to employ other people. So let's go back to the lecture. So I think you had already started on the role of aquaculture, biotechnology in aquaculture. We have looked at the areas of intervention, and we say these areas are not just unique to aquaculture. They're the same areas of intervention for terrestrial animals, or terrestrial plants, the same for the marine ones. Those are the main areas where you want some intervention. Maybe for aquaculture, the other thing is, we know we've got, what do we call it? We've got, there's this aquaculture we have. ornamental aquaculture. Probably then that's a bit different now from the rest, because this is where you're growing fish just for ornamental purposes. So probably that one is not included in this list. But this list is basically general for both marine and terrestrial, for both animals and plants. In I'm giving examples of reproductive biotechnologies. Is everyone okay? Okay, so yeah, some of the applications include for reproductive biotechnologies include one, you'd like to control the maturation in fish. If you ask most people, they want the fish to mature faster. For some fish, I think the maturation, like I think for milkfish, which is a very famous or common fish which we culture here at the coast, the maturation time is 2 years for the broodstock. So before 2 years, then that fish will not be able to spawn. So one of the targets in aquaculture is to see, can we shorten this time? Yeah, for reproductive to be faster. The other one is the triploid induction in mollusks. In this case, I don't know whether you remember in SMR 301 when we are talking about polyploidy, having more than the 2 chromosomes inside any organism, having 3 chromosomes or more. Okay, we know that animals that have triploidy or four chromosomes and going on, most of these animals are sterile, but they also seem to grow bigger. And I think some of them are actually monosex, yeah. So this is another area of intervention in reproductive technologies in fish and mollusks. Then we've also got the issue, the monosex, especially in salmonids. We also have the monosex in tilapia. Then, of course, we've got generational lines for production of transgenic fish. Transgenic fish are the fish that simply have been altered, their genetic makeup have been altered. Remember, in triploidy, you're still operating at chromosome level. In monosex, you haven't touched the genes yet. But now when it comes to transgenic fish, yeah, definitely, you have touched the genes at that time. So it's good to keep that very, very clear in your mind. Very, very clear. So what is triploidy? What is triploid induction? For example, in mollusks, triploidy refers to a genetic condition in which there are 3 sets of chromosomes instead of the usual 2. Now, for human beings, triploidy, a child who has triploidy or is conceived and starts developing and has triploidy, most of those children die before birth, or maybe shortly after birth. So they sort of don't survive. But with animals, it seems like they are able to survive. For example, we've got the triploid Pacific oyster. Sorry, I didn't write Pacific. The triploid Pacific. It is so huge. It's very big. It grows very big. It is sterile. That means it cannot reproduce. And they can thus maintain their quality year round. I'll tell you what, when you're capturing these animals, just because of the sex hormones, and they are having to reach a point where they have to start concentrating on sexual maturity. So part of the energy, it goes to the development of the sexual organs and getting ready for reproduction. And also when you bring in the hormones issue, the taste changes, the smell changes, and that kind of thing. So when you produce triploids, a triploid oyster, that means it is not going to waste any energy in matters to do with reproduction. It's going to give 100% of its energy into growth. So they grow faster, they grow bigger. And then the taste also, because they do not need, they don't need to deploy those sex hormones because they are sterile. Then again, they are saved from that weird smell that they have, or taste for that matter. So you're killing two birds with one stone. You are making them grow very, very big, and you're also making them more tasty. And this means, because there is nothing set for reproduction, you can maintain their quality throughout the year. Because what normally happens in some times of the year when reproduction is taking place, then the quality goes down or it changes. But in this case, when they are sterile, then you can be sure of your product, you can be sure of your quality throughout the year. Now, triploid oysters may also be induced by chemicals. You can use chemicals like disruptors. In the second meiosis stage, you can use very many different ways of doing it. And we are not going to go into that because that's not part of our interest for now. Then we've also got a generation of all male and all female fish. I think this one, we looked at it again in SMR 301. There was the use of hormones, the male hormones, to make sure, like the tilapia, if you want the tilapia to be all male, then when after the egg yolk stage, which is day 3 of the larvae, then you feed them with feeds are laced with androgen hormones. And because at this stage of their lives, they are sexless, they are neutral in gender, and the androgen hormones will influence them to becoming male. So about 99% of them will end up being male. And so you culture the male species. In cases like, I think, the sturgeon, a fish called sturgeon, they prefer the female to the male. It grows bigger, it is better. So they'd feed them with estrogens, which will make them feed less with estrogen, which will make them to convert to all female and with hardly any males. And this is good for production, it's good for business. Another way of producing all female or all male fish is using gynogenesis and also androgenesis. Again, in gynogenesis, you can shock the fish, you can shock the egg, so that even during mating, in the egg and the sperm meeting, then if it's gynogenesis, then you only want to preserve the female chromosomes. So the male chromosome is deactivated. And so it doesn't pass any genetic material to the progeny. If it's androgenesis and you want all male, then you shock the female, you deactivate the female chromosome. And so the progeny does not inherit anything from the father's side. And so they end up just bearing, it is there, excuse me, the new progeny, the progeny that comes up has only the characteristics, the traits from the father, and has not inherited anything from the mother. So let's look at biotechnology and nutrition. Say in aquaculture, you can intervene in the growth issue which you have just discussed. Now let's look at the feed and nutrition. Well, and we know that nutrition in fish is very, very expensive. Actually, it carries between 35 to 60% of the total cost. This figure we've been given for salmon, and the same goes for even marine tilapia. Feeding is very, very expensive. It's a very expensive recurrent cost, and yet you need it. So normally, for example, in salmon, a salmon is a carnivore. So it's a fish that survives by eating other fish. So if you have to formulate a feed for fish, for salmon, then it has to contain some some fish protein. Then what has been happening? Naturally, you go out there, catch fish, come produce fish meal, and you feed it to salmon. And so the challenge has been, is aquaculture really reducing the pressure on the fisheries? Because you're going out there and you're catching maybe 2 kilograms of fish to feed 1 kilogram of fish that you're culturing. So that has been the debate, and it's been like, no, this cannot be. So I think that it is a challenge that the aquaculturists are trying to address. And in one of the ways they are trying, or what's one of the things they are addressing, is trying to get alternative plant-based proteins. Because plant-based proteins are easy to grow. You can grow them on land, and they cost less. So they are trying with the canola meal. And canola meal is simply made from canola oil. Canola is a plant, and it's being floated as a substitute for fish meal. It costs like half the cost of fish meal. So canola meal, however, contains a substance. This substance, the phytic acid, which the salmons cannot digest. So that is the problem with canola. It is good, has a lot of protein, but the salmon cannot digest because they lack the enzyme phytase. So that means most of the phytic acid will end up being excreted, and yet you need it. You need the fish to be able to digest it and use it for as part of their nutrition. So what happens? How does biotechnology come in? So the biotechnological solutions include the canola plants can be genetically modified to produce the low phytate canola seeds. So that is one way. You genetically modify this plant and just make it produce low phytate canola seeds. I told you now, once you start playing around with genes, the opportunities are endless. Alternatively, it may be possible to use phytase supplements in salmon diets. So yeah, you feed it, you feed the fish with the canola, but you also feed them with the enzyme phytase. So that enzyme phytase will be able to digest this phytic acid. And as you can see, to be able to digest that phytic acid, and it will be useful in the body. It will be useful in the fish. The fish will be able to absorb it and use it as part of its nutrition. So the benefits of biotechnology nutrition in aquaculture, it includes that it's possible to come up with lower cost of feed for fish producers. And this will make a very, very big saving for the fish. And of course, a cleaner aquatic environment, yeah, due to less phosphorus, because if you're feeding them with canola, which is just being eaten and all the phosphorus is being excreted, then that means it's the environment will be full of phosphorus and nitrogen waste, but if they can be used better, the better. And of course, the other benefit is that you rely less on the wild caught fish, the protein, and you safeguard the environment. Now, there's another thing in fish. Probably for us, it's not so important, but in the wild fish market, this is very, very important. If you go to the wild fish markets, people go for various things. People are interested in various things in the fish. And one of the things is the color, the pigment, or the color of the flesh, the meat. For wild salmon, if you're eating a wild salmon, which is natural, their pink is their flesh is pink in color. This is a natural pigment. It's called astaxanthin. and also the other one, Canthaxanthin. They are found in planktonic algae that these fish feed on, and also cyanobacteria. And remember, these fish are carnivores. So, for example, when krill, they eat a lot of krill. When krill eats cyanobacteria and the planktonic algae, and then the salmon eat them, they end up getting these two pigments, and they make their flesh look pink. And pink is good. It makes people want to buy fish. It's one of the things that people look for when they're going to buy salmon. But now, for the cultured salmon, which are not accessing this krill, that means they are not. You're just feeding them on formulated feed. That means they are not able to develop this pink flesh. And that's something that puts off people in the market. They're like, oh no, this is not, I don't want to buy a salmon which looks white, whose flesh is white. So the aquaculture people have to think, how do we make the flesh of a cultured salmon look pink and be attractive? So one of the options, it's to feed them with dyes are supplemented with synthetic function of astaxanthin and canthaxanthin. Okay, fine, whatever. A bit, it's mouthy. The word is like too mouthy. So in order to turn their meat pink, so synthetic carotenoid pigments represent 15 to 20% of the cost of production. In order to buy these supplements, they are expensive. Okay, and you can already see it's taking 25% of the cost of production, and that's a very expensive alternative. Yeah, so and in any kind of production, you want to always keep your costs as low as possible. So are there any biotechnological solutions? Probably. So one of them is to produce plants which are genetically modified to produce this pigment. And of course, then feed them to the fish. The other one is to produce salmon that can synthesize the pigments themselves. So you put that gene in them so that they can, you don't have to feed them anything. The flesh will just turn pink. And also to improve the ability of the salmon to take up and deposit these pigments, reducing the quantities required in their feed. All those are alternatives. They are biotechnological alternatives. Probably they are still in trial, they are being tried. So the one which is maybe the most cost effective and the safest, probably it will be adopted. So what are the benefits? We already know that you'll be able to lower the cost of feeds. So definitely also lowering the cost of production. But you also be able to increase the opportunities for aquaculture feed companies to develop new products. Remember, all these things now are opportunities. So that's what I'm saying. It's like an open book. The opportunities are just out there. It's for people to take them up. So let's look at growth. So we've done feed, we've done reproduction. Let's look at growth. And in this case, basically what all this is saying is, can we be able to accelerate the growth? Can these fish or mollusks or crustaceans grow faster than they grow? For example, can we harvest them maybe like 3 months earlier or 4 months earlier and that kind of thing? So all these, you can use recombinant somatotrophins. This is both bovine placental lactogen to try and improve their growth. And this is highly effective and it can be used to control growth via transgenesis. But it is also very, very severely regulated and there must be reasons. Okay, The other thing is to insert appropriate DNA constructs in the genes of fish to promote faster growth. You want to put a growth factor or something that will improve the growth hormone in that fish, so that, or whatever organism it is, so that they can grow faster and reduce the production time, which will also save on the growth. So what are the benefits? If you're able to culture fast-growing aquatic animals, you can increase fallowing. And this protects water quality, marine sediments, and aquatic health. Fallowing is you use less time to grow or to use the marine waters. So you give some rest. You give the water some rest, a break. Instead of maybe using it year round, if you're able to grow your fish faster, you can give the waters a break for them to recover their health. Then you also reduce cultivation time. Of course, the supply can be increased without increasing the use of coastal water. So I think the idea here is give the marine waters, the coastal waters, a break. Okay, instead of you're always using them to produce your fish faster, give them a break, let them clean up, let them renew, regenerate, and then come back later. Then you also get fish that are growing very fast, also have very high conversion rates. That means fewer natural resources are required to produce them, thus enhancing sustainability. And I see this is going to be a big one. And then improving the economics of aquaculture so that land-based systems become economically viable and competitive with ocean pen systems. Yeah. If you remember, when you're using the technology, not only are we using it in the marine system, but we're also using it on the land-based system. So is there, can the two match? You're growing, maybe you want to grow your maize at 3/4 of the time that you normally use. If you can also grow your fish 3/4 of the total time that you use, then the two you can say the two are going hand in hand. Those who are economically viable on land, and also the marine systems have also not been left behind. Okay, so now let's look at health, biotechnology, and health. We know that DNA and RNA and other diagnostic tools are providing faster and more accurate ways to detect and identify fish and shellfish diseases or disease agents. And this is important because you get more effective monitoring and management of health, and you're able also to intervene very early. So early detection means early intervention. So that means if you address the problem early enough, then you have the reduced use of drugs for treatment, antibiotics, and everything. Then you're also able to remove the potential disease-related barriers to international trade in aquatic animals. Then there's also the manufacture of pharmaceuticals and nutraceuticals. Now, chemicals produced or found in marine organisms have been shown to have a wide variety of applications, from pharmaceuticals in human and animals to everything, name it. They have been used as antibacterial, antiviral, anti-tumor, name it. Nutraceuticals constitute a variety of substances used in food supplements and natural health markets. I'll tell you what. You know, I'm sure, I don't know whether we went with you to Kiboyoni to visit the aquaculture farms, seaweed farms. But I just got talking to some people in Nairobi, and they were telling me there's a product, it's this health what do you call it? The health supplements, the people who sell health supplements. They were telling me they are buying seaweed from Tanzania, and they were just coming, they were bringing it over to Kenya and packaging it, and they would sell a kilogram of the powder, of the seaweed powder, at 2,000 shillings. And I was like, which species of seaweed are you looking for? And they said, oh, the red seaweeds, Spinoza. And I'm like, we have that in Kenya. Why do you have to go to Tanzania? Oh, we didn't know, and that kind of thing. And in my heart, I was saying, that's an area, that's something that nobody is bothered about in Kenya. And that's an area. The seaweed, actually, they call it sea moss, has a lot of nutrition, a lot of benefits, a lot of nutrition benefits. I mean, if you see something going for 2,000 shillings for half a kilo, then that is something. So, no, for a kilo, I think. So when you talk about nutraceuticals, I think the one which is right here, what you can call a low-hanging fruit in Kenya, is just the seaweed. And yet nobody is looking into it. So probably some of you will start thinking about it seriously. I don't know much about it, but probably, how do you gain knowledge? By reading, by talking to people. Okay, let's move away. There's also industrial chemicals. And of course, our best example is carrageenan that is extracted from seaweed. Now, this is for industrial purposes. And again, in Kenya, nobody is using our carrageenan. We have to export it all the way. The women are trying to make soaps. They are trying to make all these other things. But we are so slow in taking up this, unfortunately. Okay. There is also a variety of other chemicals used in both food processing and other manufacturing processes that can be used. Then we have others, include biofouling. If you know what fouling is, if you've ever looked at a boat, the boat that's always in water, after some time, you'll see some animals or some organisms have started growing on the boat. You get barnacles, you get all sorts of some mollusks hanging onto the boats and living there and that kind of thing. And this is very irritating to the boat owners. So mostly what they paint their boats. Maybe you didn't know this, but boats, if you see the painting on the boat, that paint is not normally the normal paint. It's called marine paint. And that marine paint is supposed to keep off these falling agents. If you look at most boats after some time, they're just full of barnacles, and they have to pay a lot of money for people to clean up those boats. Let me look whether the students from Coast are here. Where they go to the beach. Omar, are you in? Is Omar in? Okay. Don't know where Omar went. Who was? Okay, I don't know. Yusuf Omar, are you in?
Yes, I'm in.
So do you know what I'm talking about when I'm talking about falling? Omar.
Pardon me, when you talk about?
You talk about falling. Have you been to the sea?
Yes, I've been.
Have you looked at the boats, the sides of the boats? What do you normally see? Omar, come on, open your... Talk to me.
Oh, they are usually held by certain floaters and anchors. They are usually held by certain floaters and anchors.
So what do they do? What do the fishermen do after some time? Omar, you're off again.
No, I'm still on.
No, you had muted. So what happens? To get rid of the animals, what do the fishermen do? Or the boat owners? Okay. No help.
I'm not sure.
You're not sure. You guys don't go to the sea. You don't interact. I don't think you're observant, you guys. You don't take time to observe things. They have actually to look for people to scrub the boats clean, to get rid of those algae and all those things, the barnacles and things that are stuck to their boats. And then after some time, after a year, they dry dock their boats and they paint them again. After they cleaned up everything, they paint them again with marine paint. So falling is a real big issue in fishing, in all those that are using boats. So what they are looking for now, in order to limit anti-falling, they are trying to see, can they use bacteria to try and deal with this problem? Because some bacteria have their own natural systems of anti-falling to make sure that other animals don't come and ride on them. So so some of these anti-falling products can be extracted from fish, the mucus of fish. Fish also, I mean, we know once you hold the fish, it's slippery. That is for it is a way of anti-falling that other creatures are not coming to hang on to them. And starfish also, for example, they prevent sessile marine animals such as barnacles from attaching to the boats because they eat them. You know, all these things. So they are looking into how can we use these animals to also develop anti-falling products for the boat. And then from there, we've got marine pollution. I think we looked at that, so I'm not even going to look at it. We looked at what to do with marine pollution and everything. So as I wind up, marine bioprospecting, what exactly is this? I'm sure you've heard of it before. So what I'm going to do, I'm going to show us a video, and then I will explain. This one is called Marine Biodiscovery. Can you all see? Can you see my screen?
Yes.
Okay.
Biodiversity in the oceans is enormous. The deeper you go, the stranger life gets, and the more likely you are to discover new life forms. Marine biodiscovery is the process of discovering new products and processes from marine biodiversity. Hi there, my name is Marcel Jaspers. I'm Professor of Organic Chemistry at the University of Aberdeen and Director of the Marine Biodiscovery Centre. I've been working in the field of marine biodiscovery for the last 25 years, and I'm now using my knowledge to help the United Nations develop a new law to conserve and sustainably use biodiversity of areas beyond national jurisdiction. Today, I'll explain to you what we do in our lab, how we do it, and why. My group and I welcome you to our laboratory to see what we do. The work we do is mainly most of these work in very new and unusual ways to treat disease. Besides pharmaceuticals, we also try to discover enzymes that do industrial processes in a cleaner and greener way, or to discover new nutraceuticals or cosmetics or personal care ingredients. The marine biodiscovery process can be divided into a number of steps, starting with the collection of an organism or sediment and ending with the discovery of a new molecule. The process of taking that molecule from the bench into the clinic can take up to 20 years. We start by collecting organisms such as marine invertebrates, in particular sponges, sea cucumbers, sea mosses, and soft corals. Nowadays, we are more likely to collect deep-sea sediment using a grab or coring device and use this to obtain microorganisms that might produce interesting molecules. Once we have the deep-sea mud, we ship it back to the lab for strains of bacteria or fungi. One mud sample can potentially contain thousands of different strains of microbes. Once we have a pure microbial strain, we will grow a batch of it in liquid culture for chemical extraction and biological testing. Once the bacteria have grown for a few days, we extract the compounds they have made using solvent extraction. The extract can contain a mixture of hundreds of compounds. The next step is a cycle of biological testing and compound purification. It can be tricky to discover the one biologically active compound in a mixture, and we use a range of methods to achieve this, such as solvent-solvent fractionation and chromatography. We test extracts, fractions, and pure compounds against a range of diseases, typically using cell-based assays. In this, we treat, for instance, cancer or bacterial cells with our materials and see if they kill them effectively. Most of this work is automated, as often thousands of tests are necessary to discover a few biologically active compounds. Once we have isolated a pure, biologically active compound, we need to determine its chemical structure. This is done using spectroscopy, which tells us the mass of the molecule and the connections between the atoms in the molecule. The instruments to do this are highly sophisticated, but the methods are well understood. A new biologically active molecule to a marketed drug is a long road. First, the molecule may need to be modified and produced in larger quantity. This can be achieved most commonly by chemical synthesis or fermentation. After additional cell-based testing and physical property measurements, the compounds are tested in animals before being tested in humans. Human clinical trials can take between 2 and 10 years to complete before a drug can be marketed. Many marine-derived molecules are used to treat cancer. Yondelis, derived from a Caribbean marine invertebrate, took almost 20 years to develop. It is used to treat a type of cancer for which there was no treatment for the previous 25 years. It is produced using a method in which a fermentation product is modified chemically to synthesize the molecule. Therefore, it's a sustainable process. The treatment dose is so low that it's only 1 milligram per treatment cycle. If you want to imagine that, imagine 5 grains of sugar on your hand, and that is 1 milligram. The most remarkable thing about marine biodiscovery is that the drugs we discover have unique ways of hitting the disease. Drugs with new mechanisms of action are necessary to overcome resistance that is common in cancer and infection. Thank you for coming to our laboratory today. I hope you learned something about the process of marine biodiscovery and found it interesting. Thank you.
Wow, thank you too for, I thought that was also a nice presentation. Again, opening doors for some people and saying, yeah, for some it's just a matter of creating knowledge, which is okay. I think for some it's beyond that. Some probably these are some of the things they want to pursue, which is good. So marine bioprospecting, as we have seen, is usually includes collecting the marine biota for natural product screening, and we've been taken through that screening, although I don't expect you to remember all that. And traditionally, what has happened, 1 out of the 10 to 20,000 molecules that are extracted from terrestrial microorganisms would take almost 15 years, and I think that guy has said up to 20 years to get to the market at the cost of about 800 million. So what happened, most pharmaceutical groups abandoned their search for new drugs derived from natural substances. I think the opportunity for encountering successful candidates is quite high. And just because of, number one, the extreme environments in which some of these organisms live, and the fact that we also don't have a lot of knowledge. So, and I think also the biodiversity is quite high. Anti-cancer agents for marine organisms have estimated to be about 1 billion a year, $1 billion. So that's quite huge. So as I think that person said, the presenter said that this is still in the offing, but there is a lot of interest and it's happening. Probably the methods are going to get better. Probably the process is going to get shorter and better. I don't know. And finally, I think this is going to be a reality in most cases. So I am thinking that that's enough for today. It's enough food for thought, enough material to go mull over, think through it. And then I think we meet on Wednesday. I will let you know. First of all, let me read let me look at the papers you have chosen, because I have to read them. I prepare your card. And from there, I will let you know the exact time we'll meet on Wednesday so that we can do the card. And then the other 2 units, this one and the one we taught in the morning, I'll also be telling you when we are going to do the first card. So I think I'm giving you time to go and read for that card, get ready. Don't only read the material you chose, read other related materials. So, and I'm going to find out whether you read other materials because I'm going to look at what you're quoting. So I think, yeah, we've come to the end of our lecture. Make sure you write your name if you haven't written it. And let's meet on Wednesday. I'll give the specific time to the class rep. Have a good evening or afternoon for that matter. It's still early afternoon, so have a good afternoon. Bye.
Bye.
Bye.